2016. Our first speaker today talking about short motifs is Shobhit Jain, who's a PhD student at Gary Bader's lab, and he'll be talking about predicting high confidence PRMs, or SH3 domain peptides mediated protein-protein interactions in yeast.
Okay, so basically I will give you a brief introduction and then discuss some of the results of the project on which I'm working on, and then finally proceed. So there are many intercellular signaling processes which are mediated by interactions involving PRMs such as SH3, WW, or PDZ domains. Now specifically the SH3 domains, they bind to small linear protein-rich regions in animal proteins. There are many experimental methods such as peptide chips or phase display which can be used to identify the short motifs which bind to these domains. So in my project, we use phase display data. We use phase display data and then we try to predict protein-protein interactions involving these SH3 domains and the short peptides which are generated on phase display. Now, phage technology actually permits us to display extremely diverse libraries of peptides on the surface of phage particles. Now, once created, these peptides can be selected by immobilized domains, in our case SH3 domains, and once we have a bunch of peptides for a particular SH3 domain, then we can use those peptides to predict protein-protein interactions. So our straightforward approach to predict protein-protein interactions using domains and peptides is to construct a position rate matrix using the peptides generated from the phage display experiment, then scan the whole proteome. Using some thresholds, we can collect the number of hits, and these are our potential targets. But the problem with this straightforward approach is that you get many false positives. For example, the PWM does not capture information such as the binding site if it is buried inside the 3D structure of a protein. And that type of information is not there in this PWM or the PWM scanning. For example, the protein containing the SH3 domain and the protein which has a peptide, they might not even be at the same location. One of them might be a membrane protein, another one could be localized in nucleus. So the likelihood of them to interact is very less. So these type of things, they are not captured by this straightforward PWM scanning approach. So we need some extra information. And this information is coming from different biological data sets. So the first step is to collect this biological information which helps us in predicting high confidence PRM-mediated PPIs, and then to integrate all this information to construct a single model which can do the prediction. Now, so the biological information which we have collected to do these type of predictions, we have divided this into two sets. The set one, which we call the peptide features, they basically gives us a high confidence binding site. So we use surface accessibility, the conservation of the peptide, structural contact between the peptide and the domain, and the disordered region information to predict high confidence binding sites. Another set of features which we are calling as protein features. They basically tells us whether the two proteins are likely to interact by terms of their proximity, whether they are in the same location at some point in time, or they belong to the same biological process, and so on. So we combine these features to make these predictions. Now we'll quickly go over these features of how do we process information using these features. So in case of disordered region, we know that SH3 domains, they bind to proline-rich regions in a protein. Now these prolines, they tend to accumulate in the disordered regions of a particular protein. So we use disoccurrent to find out the disordered regions in a protein, then use that information to score the peptides which have been generated by using the PWM scanning. Similarly, the second information is surface accessibility. So if the peptide is present on the surface of a protein, then only it will be accessible for binding. If it is buried inside, then the binding between the SH3 domain and the peptide will not take place. So we use SABLE to predict the RSA values, and then estimate a surface accessibility score for that particular peptide. Then we'll look at the conservation of that peptide among different yeast species. So as these interactions they are involved in signaling, so the hypothesis is that the peptide should be conserved. There should not be mutations, or it should be conserved as compared to the remaining sequence. So we use in paranoid, and then we do the alignment of sequences by using MFTP, and then use this information to calculate a conservation score for that given peptide. And the fourth information which we are using to identify the binding site is the structure contact. So we use known 3D structures of SS3 domains which are complex with peptides, and then use this information to assess the binding potential of our query SS3 domain in peptide. For this we are using 6 these SS3 peptide complexes from 3D, and then we convert the contacts between the peptide and domain into a score, and finally calculate a single score for a particular peptide which has been predicted as a potential interactor using PWM scan. So these 4 features, they will tell us whether the binding site or the peptide which we have identified is a high confidence or not. Then we have a completely different set of features which are protein features, which are working on the whole protein sequence. So for example, cellular location, biological process, and molecular function. So for a true interaction, the two proteins, a true physical interaction, the two proteins should come in close proximity to each other. So for a true interaction to take place, these two proteins should be in close proximity to each other. Or they should be part of a same biological process. Then only the interaction is more likely to take place. This information is present in gene ontology. So gene ontology has it's a controlled vocabulary which contains 3 ontologies. One is cellular location, biological process, and molecular function. And by using this ontology, you can get the information whether the two proteins are likely to be in the same place or not, or whether they could be part of the same process or not. So we use semantic similarity measure called topological clustering semantic similarity to identify the relationship between two proteins given a gene ontology. So by using the semantic similarity and gene ontology, we generate 3 scores. One is for the cellular location, another one for biological process, and the third one is for molecular function. These 3 scores will tell us whether the two proteins are likely to interact with each other or not, based upon this information. Then we look at gene expression. So cell is optimized to co-express genes which are functionally related. And if they are functionally related, then they are more likely to physically interact with each other. So what we do is we combine correlation coefficients from 86 different gene expression data sets and generate a single score which tells us basically whether the two proteins are likely to interact with each other or not based upon the gene expression information. And the last feature which we are using in case of proteins is sequence signature. Now, functionally related proteins have similar domain composition. So we look at the domain composition of the two PPIs and use that information to calculate a score for our query proteins. Now once we have all this information, the next step is to integrate everything. So the first step is we are starting with phase display experiment of the peptides which we get from the phase display experiments. We generate a PWM using those phase display peptides and then scan the whole proteome. This gives us a bunch of hits. Now for every hit which we get using the PWM scanning, we calculate a probability whether the interaction will take place or not based upon 2 different models. One is the peptide and another one is the protein model. So the peptide model, we integrate these 4 peptide features using a night vision classifier. In case of protein model, we integrate the different features again using a different night vision classifier. We get the 2 probabilities based upon these 2 feature sets. Then we finally combine these two probabilities to predict whether the proteins are likely to interact with each other or not, and which is the high confidence binding site present inside this protein. Okay, so this is the analysis of the feature set which we are using. So the thing is whether these features are giving us any information or not. So it's as it clear from the figure that all the features, whether they are in peptide or protein, they are helping us in predicting protein-protein interactions, or giving us high confidence protein-protein interactions. But if we combine these features, then obviously the score gets higher, which is the black curve. Now, if we combine both the peptide and the protein classifiers, then we again improve the accuracy of the prediction model. So, for a particular cutoff, for example, for a particular p-value of -5, you can see that the combined classifier outperforms both the protein and the peptide classifiers. Okay, finally, to summarize, so we developed an algorithm which helps us in predicting PRM-mediated PPIs. It combines the binding site information, which are the peptide features, with the protein features using Bayesian modeling, and the combined model outperforms the individual models. Now, we used phase display data from Dominican's paper and predicted roughly 357 novel interactions, which with approximately 480 binding sites. Okay, so finally, I would like to thank my supervisor, Gary Bader, my committee members Alan, Mike, and Quaid, and I would like to thank Dave and David for providing us with the phase display data for this. Thanks.
Thank you. I'll leave a couple minutes for questions. Maybe Alan can set up in the meantime.
Do you have a plot that can show the contribution of each of the items that contribute to your model? Like how much each of the
features?
Yeah, each of the features.
So the plot, the curves that showed how each of the feature contributed towards. So all the features they contribute towards predictions. Some of them the contribution is low, for some of them it's very high, but all of them.
So what's your next step?
So the next step will be to use the phage display experiment data from humans, which was recently generated by Dave's lab, and do a similar thing with humans and include more features, because in case of humans, we also have protein expression data. So we will be using protein expression data plus a couple of other features and try to improve the predictions.
I noticed that the gene expression was actually quite low in terms of improving the prediction, and maybe that's not really unexpected, just because you could have continuous expression in one case and sort of.
Yes. Yeah. So we actually did an experiment where we compared the gene expression and the protein expression, and protein expression in that gene performed much better as compared to gene expression in case of humans.
But two genes don't need to be co-expressed to interact.
Yes. Yes. That's true.